functionalities to dCas9 to control transcription, edit the epigenome, or image the genome, or mutate the genome. So I think that today is a really sort of nicely matched set of talks, I think. Some of what I'm talking about will be a segue into some of the things Mike's talking about. And I'm going to talk about sort of single gene approaches, and then you can imagine scaling a lot of these technologies up to ask additional questions about how the genome controls function of various cell types. So this is the same outline you saw yesterday. We have a lot of slides, so I'm going to skip through the intro. So initially, as I said, we used dCas9 in bacteria just by targeting this protein to specific sites along the genome. We can inhibit RNA polymerase elongation. This unfortunately does not work so well in human cells, and so we've taken to appending additional domains to more robustly control transcription. And so our goal early on, once we realized that dCas9 alone didn't perturb transcription by much in human cells, was to ask whether we could append domains to either silence transcription or activate transcription to dCas9, so that in essence we have the platform shown on this slide, where we have an RNA-guided DNA binding platform with which we can fuse a variety of effector domains and easily target these effector domains to arbitrary regions of the genome to enact some function. And so we one of the reasons I talked extensively yesterday about the historical perspective of synthetic biology and controlling transcription was we moved very quickly early on because we could take a playbook from what had been done and just repurpose it over to CRISPR. And so I showed you ways that you can silence or activate transcription yesterday using various domains that have been defined over the last 20 or so years. And so we, not being very creative, took a handful of those domains and appended them to dCas9 and asked whether we could control transcription. And so this is one of the earliest experiments that we did, where we took this well-characterized CRAB domain that I had shown you yesterday. This is a scaffold that recruits additional enzymes to change local chromatin marks. And so we appended this domain to dCas9 and transfected or infected reporter cells that expressed a simple GFP reporter with a guide targeting the reporter and this CRAB dCas9 fusion domain, and then just asked a very simple question a few days later whether we could turn GFP expression on. Sort of exactly the same experiment as I showed you in E. coli, but here we're using human cells, and we've got some additional vector domains. So this data is shown on the right, where there's a FACS plot looking at human cells that express either this GFP reporter, and then we can compare those cells to cells that express no GFP, or if we target the GFP reporter with an sgRNA that targets this dCas9 CRAB fusion protein to the reporter, we can see that we can pretty robustly turn transcription on. So these two histograms are largely overlapping, so express the this RTTA, or this or activator protein, you induce venous expression. And then if you co-express DCAS9 targeting these TET sites, we see that we can turn gene expression off. So this is illustrated here. We don't go all the way back to baseline, but it clearly illustrates that we can use DCAS9 and eukaryotes to compete for a transcription factor binding site and control gene expression, sort of functionally mapping sites in an artificial way, functionally mapping sites along a promoter which are required for controlling gene expression. So this is I think sort of an underexplored use of DCAS9, but it has a lot of potential for mapping regulatory regions, sort of in a very fine way. So our ability to control transcription is only useful if it's specific. So if you have, doesn't matter how robust your ability to control transcription is, if you're deregulating many genes across the genome, that's not going to be a very useful tool for understanding how biological systems work. And so I'm not presenting these experiments in temporal order, but as we saw, we could use these DCAS9 crowd fusions to edit that
cannot